Now we're going to move into talking about the ionization of amino acids. I've touched on this a little. I've noted that there are these special R groups that have charges on them, and also the fact that amino acids, when they are neutral, they have this negatively charged carboxylic acid and this positively charged amine on them. So let's talk about how and when amino acids ionize when they form charges, and why it's important. So all amino acids contain at least 2 protons that are ionizable, that will be charged. And they each have their own pKa. Remember that pKa is related to the acidity of the particular molecule. So if we focus on the part of an amino acid, so not an amino acid as a whole, right now we're going to focus on parts of it. The first part that every amino acid has is the carboxylic acid, and this functional group is very acidic. This functional group has an acidic pKa, and that means that it will be protonated only at a very low pH. So COOH will go to COO minus and give away its H plus. So again, it has a very acidic pKa. What does this mean? Remember that a low pKa, an acidic pKa, means that this acid is good at being an acid. It dissociates very well. So that means that COOH, carboxylic acid, dissociates really well. It even has acid in its name, right? That means you normally are going to find COOH as COO minus because it has dissociated. Normally, in most cases, it's going to do a good job at dissociating, getting rid of its H plus. It's very acidic, and it will be in the form of COO minus. This is why we normally write our amino acids with that COO minus on it. The second group that all amino acids have that has a proton on it that is ionizable is the amino group. This group has a very basic pKa. It's not a good acid. That means that it generally will stay protonated even into a very high or basic pH. To write this equation, NH4 plus goes to NH3 plus H plus. So here's this group. Note that the version that has lost its NH or its H plus here is on this side of the equation, on the side where it is dissociated. But we just noted that this group, this amino group, is bad at being an acid. It's bad at dissociating its H plus. It has a very high pKa, which again means that this is not going to dissociate in normal conditions unless you are pushing it to a very, very basic pH. You have to push it to a point to force it to get rid of its H plus. So that means you normally will find this group in this formation, the NH4 plus formation. Again, that's why we write those amino acids with the NH4 plus. So again, remember that pKa, we're going to be talking about pKa's a lot. pKa's are a measure of acidity. They tell you how acidic, how good at being an acid a group is. Now, remember a hint, if something is a functional group that has the word acid in it, that usually means it's good at being an acid. And that means, again, a reminder that it will dissociate. It will get rid of that H+, meaning it will be left with a negative charge at very low pHs. So here's our carboxylic acid. You can see it will form the COO minus, the negative form, at a low pKa. If you have a high pKa, if you're a group with a high pKa, you're bad at being an acid. That means you will hold on to your proton. You'll hold on to it until you finally are forced into a very, very basic pH in which the base will finally pry away your proton. So remember what pKa means. It's telling you how good of an acid something is, how good it is at getting rid of its H+. So for amino acids, the carboxy group, that COO minus, that COOH group, is a better acid. It's much more acidic than just a normal carboxylic acid even. So in this case, the carboxy group on an amino acid is actually even better of an acid than just, for example, normal acetic acid. So a COOH group on an amino acid like glycine actually has a pKa at closer to 2.3. That means that only at pHs below 2.3, so something very, very, very acidic, will it still have that H on it. Otherwise, it will be in this form with the COO minus. The amino group, the NH2 group on the amino acid, is just slightly less basic than a normal amine, like a methylamine. You can see here's an example of methylamine, and its pKa is around 10.6. The amino on an amino acid is just a little bit slightly less basic than that. Its pKa is going to be around 9.6. So remember, this is in general for most all amino acids. The carboxylic acid group on an amino acid is down. Its pKa is very low, around 2. And the amino group, that NH2 group, has a pKa that's very high, around 9. Which means that this whole other range between here and here, between those two pHs, the amino acid exists like this, with the COOH having given away its proton, and it is in the COO minus form. And the NH3 plus, it hasn't yet given away its proton. It's in the NH3 plus form. So again, only at a low pH can an amino acid exist in a positively charged form. So at a very, very, very low pH, your COO would have its H on it, and you'd have an NH3 plus. This is at a low pH, very, very low. This has not dissociated yet. And at a very, very, very high pH, we now have dissociated this and this. So we would have a negatively charged amino acid. Between these two extremes, we are going to have this form with the negative and the positive charge on it. This is a neutral molecule that has two charges on it, which we call a Zwitter ion. So again, it's an ion that has a positive and a negative charge on it, but overall is neutral, right? Those charges cancel each other. So overall it's neutral, but it has a positive and negative charge on it. So amino acids tend to exist in this Zwitter ion form. Now, when we look at a plot of an amino acid's pKa, the Zwitter ion form is in a special area that we call the pi. This stands for the isoelectric point, the point in which it is a neutral molecule. For a normal amino acid that does not have a charged group on it, it does not have a group that is ionizable, we say. So something like glycine, something that just has a carboxylic acid and an amine group on it, and it has just like a normal R group with carbon and hydrogen, no charges. This point where the amino acid is a Zwitter ion is going to fall directly between the pKa of the carboxylic acid and the pKa of the amino group. So again, most amino acids, all amino acids are going to have these two pKa's, one for the carboxylic acid group and one for the amino group. So they'll have two areas, two buffering areas, two pKa's, and they'll have a curve that looks like this. If you want to calculate this isoelectric point, this point where the amino acid is neutral, then you take each of the pKa's and you find the average of them. So it would be pKa1 and pKa2 divided by 2. So find the average. That will give you the isoelectric point. That will give you the number, the value of where this amino acid is exactly neutral in its Zwitter ion form. So this is the point where the amino acid exists as its Zwitter ion form between those two pKa values. This form is important to note. The reason we want to know where is this pi, at what pH does it exist that this amino acid is perfectly neutral. Because when an amino acid is perfectly neutral like this at the isoelectric point, the amino acid is not very soluble in water. It's the least soluble in water. Why? Because it's neutral, and we already know that things with charges on them are better soluble in water. And it will not migrate in an electric field. And that's important when we start talking about some of the methods for looking at amino acids. So as biochemists, to be able to identify different amino acids, this point about being able to migrate in an electric field is important. So based on what I've just told you, every amino acid has a pKa for its carboxylic acid group and a pKa for its amino group. So it at least has two pKa's. Every amino acid has at least two pKa's. And every amino acid, thus, can act as a buffer. Remember that buffers prevent change in a pH close to its pKa. So each amino acid has two ranges where it can be a buffer. For example, with glycine could buffer around its first pKa, which is at 2.34. So it would be a good buffer first at a pH range of around 2.34. And it could act as a buffer at a pH of around 9.6, where its second pKa is. So again, those highlighted regions where the pKa of glycine is, again, this is for the pKa for its carboxyl group. It's at a low pH. The acid is at a low pH. And the amino group, which is at a high pH. Those areas are its two buffering regions. Now, one thing I noted is that this is for all amino acids, but some amino acids have side groups that also have charges on them. They also are ionizable. And this is going to change where that pi is, and it's going to change what its titration plot looks like. And that's because there are extra groups on there that also can lose a hydrogen, and so they also have a pKa. They also can act as an acid and dissociate. So these titration curves are a little bit more complex. I'm going to show you an example of one of these titration curves, but know that there are a few groups, and we'll go over them, that have these ionizable side chains. There's going to be a few special amino acids that have an extra pKa group on them. One of those examples is the amino acid histidine. Histidine, remember, has a charged group on its ring, this NH right here. Now, this particular group has its own pKa. What we're looking at here is the series of when histidine goes through different charge states by each acid dissociating a proton. You can see that in its initial state here at a very, very, very low pH, our carboxylic acid has yet to dissociate its proton. And both the NH group here and its normal amino group also are charged and have their proton attached. At this point, the total charge on this molecule is +2 at this particular pH. Once we get a little bit higher in pH, the first proton that will dissociate, because it's very good at dissociating, is the one on this carboxylic acid. And we know that because its pKa is 1.82. It has a very low pKa. That means at a very low pH, it will get rid of its proton. At this point, the total charge of the molecule, you can see, is now +1. That's because there's a negative charge here, positive charge here, and a positive charge here. So in total, the charge is +1. So we've gone through the first zone, the first pKa. Now this molecule's second pKa is actually for its extra side group. And that is usually called the pKr. That's the pKa for the R group, for the side group is all that means. You see that representation. And it happens at a pKa at 6. So around pH of 6, we will then get rid of the hydrogen on that R group. So notice the hydrogen is gone here. So it's not charged anymore. And we still have our hydrogen gone here. So in total, our charge is now 0. And the last pKa for this molecule is very high. It's at a pKa of 9.17. So around a pH of 9.17, we will lose that final hydrogen on the amino group of the ring, of the amino acid. And for this amino acid, that means that when we lose that final proton from the amino group, the net charge that is left is -1. Notice that both these groups now are neutral, and we have a negative group on the histidine. This series of losing protons usually goes in this order. You will have the carboxylic acid on the amino group, the carboxylic acid that is innate to the amino acid will first lose its proton. The second group to lose its proton will be the R group. And the third group to lose its proton will be the amino group that is, again, innate to the amino acid. Now, for a molecule like this, you'll notice that the pi, the isoelectric point, is actually in a different place. And that's because now we've added in an extra group with its own charge that dissociates at a different time. The isoelectric point always marks where the molecule has a charge of 0. For this molecule, you can see that that happens between this point and this point. And that is where the pKa for the R group is, and the pKa for the amino group is. So the pi for a molecule with multiple pKa's always exists where the charge is 0, and it's going to be the average of the two pKa's between that point. So for this particular molecule, that's between this pKa and this pKa, and so it is the average of those pKa's. That's where you'll calculate the pi. Remember, again, for all other molecules that are not ionizable, it's very simple because the pi only exists between two pKa's, the carboxylic acid and the amino group. And so it's just the average of those two pKa's. When we have a ionizable group present, you have to think about, for that particular molecule, when the molecule is going to be at a charge of 0, and it will be the average of the pKa's between then. So again, here's where it is at charge 0, and so it's an average of this pKa and this pKa. So again, to calculate a pi on a amino acid or any structure really that has these multiple pKa's, you're going to look for when the particular structure carries a 0 charge. So you have to think about the order in which protons are taken off, and then identify the pKa values on each side and take the average. So last to note, I'm going to put up this table, and I suggest that you reference it. There's also a different form of this table, but it has the same information in your book as well. This lists each amino acid and its pKa's. So every amino acid has a pKa for its carboxylic acid group. That's this first pKa. This is the pKa for the carboxylic acid group. And that's for every amino acid. Essentially, that's the pH when the carboxylic acid will get rid of its hydrogen. The second pKa is the pKa for the amino group. So again, that is the pKa at which that NH3 plus will get rid of its proton, its hydrogen group. And that's for every amino acid. So every amino acid has those two pKa's. There are a total of 7 amino acids that have ionizable side groups. And you can see any of them that have an ionizable side group, an R group that can be ionized, it is listed here. So these 7 have an R group, an additional group with an additional pKa. And you can see they vary in their pKa as far as the magnitude, the charges. So something like glutamate has a R group with a pKa that's pretty low. It's at 4.25. But tyrosine has a R group that has a pKa that's pretty high. It's at 10.07. So again, these tell you at what pH along the order that particular group, that R group, will dissociate its hydrogen. So I implore that you look at all of the structures, especially for these 7 additional groups. Look at the hydrogen that will dissociate and compare it to the pKa on this chart. Think about when along the scale of pH it will get rid of its hydrogen and what will happen to its charge then.